Hello and welcome back. We are on to the second video of cardiac physiology. In this video, we're going to focus on the contractile cells. So we just got done talking about the conducting cells for the intrinsic conduction system and the ECG. Now we're going to take a look at the contractile cells and their role in the actual contraction cycle, the cardiac cycle of when the muscle cells are responding to the electrical signals that are being sent via the intrinsic conduction system. So first, we're going to take a look at the action potentials of contractile cells. So we have to remember when we're taking a look at heart muscle, it's all cardiac muscle cells. So if you remember, a cardiac muscle cell was kind of branched. It had striations, right? Because it was striated. There's probably a nucleus in there, but it was also connected to its neighboring cells. Maybe I'll grab a different color. Via gap junctions, right? So we're called intercalated discs. So when we are talking about the contractile cells, these are the cells that function in contraction, right? The muscle cells get shorter. The conducting cells, those were the unique special cells that were part of the SA node, the internodal pathway, AV node, AV bundle, Purkinje fibers. They do not contract, they just spread the electrical information. These are the contractile cells, the one that actually responds to that electrical impulse that's coming from the conducting cells and then actually causes the muscle cells to get shorter because of your sarcomere, all that muscle physiology stuff we learned back in chapter 10. Now, again, if we are taking a look at this, it's an action potential kind of idea, very much like we saw with the SA node in the pre-potential. I'll draw a reminder of our regular action potential in a neuron. Not super great, but there it is. And again, the numbers are a little bit different. So for these contractile cells, their resting potential is -90, where in a neuron it was -70. But again, the numbers aren't super important this time around. It's more of the pattern of why it's different if we're comparing a nervous action potential with the cardiac muscle action potential. So the stimulus shows up, we depolarize, right? So those are probably the voltage-gated sodium channels opening, rapid depolarization at step 1, right? So cause, sodium entry, 3 to 5 milliseconds, close it or ends with the closing of the voltage-gated sodium channels at the +30, all right? So that's very similar to what we've seen already. Here's what is a major difference between a neuron action potential and the contractile cell action potential. Take a look at this phase 2. This is called the plateau phase. What causes it? Well, shortly after that sodium channels close, no more sodium's coming in, calcium channels open. These are called slow calcium channels. Calcium is positively charged. It has a gradient of wanting to move into the cell. So when the sodium channels close, no more sodium's coming in, but now these calcium channels open and they stay open for quite a long time. And when they start to close, they close slowly, slow calcium channels. So this duration is a really long time, 175 milliseconds, which is crazy compared to a nervous action potential is done in like 5 to 10 milliseconds. 5 milliseconds is, 175 milliseconds is a forever in action potential time. So during this time, we are in absolute refractory period. So sodium channels are closed and inactivated, so we cannot generate another action potential during this whole, 200 milliseconds worth of time. Then we get into the relative refractory period, step 3. So calcium channels are finally closed. We get the opening of the voltage-gated potassium channels. Potassium rushes in, and we get our repolarization. We are now in the relative refractory period because the sodium channels are back to normal. And then our sodium-potassium pump will actually get us back to our resting state. Okay. So why does it take so long? Why do we have this extended plateau phase in cardiac muscle cell action potentials compared to, say, like a skeletal muscle action potential or a neuron action potential? Well, if you remember back in chapter 10, we talked about things like summation, like TREP and wave summation and incomplete tetanus and tetanus. And we had skeletal muscles, the neurons could fire so fast that those skeletal muscles could not rest before, they could not relax end their contraction before the next signal came in, and you could get a tetanic contraction and your muscles would just be totally tight and you couldn't relax them. Well, in a heart and skeletal muscle, I mean, that's dangerous, especially like with the disease tetanus, your diaphragm contracts and you can't breathe, you can't exhale. No, if it contracts, you can't, exhale because you won't be able to relax. But if your heart was able to do tetanic contractions, your heart would contract and it couldn't relax. How good is a pump if it can't pump, if it can't go through contraction relaxation cycles? So this is like a fail-safe. It is a physiological prevention for tetanus, because take a look what happens right here. Before the relative refractory period begins, the muscle that's responding to this, the muscle contraction, will always be able to relax before relative refractory and resting begin. That wasn't the case for skeletal muscle. You could keep firing and you could keep telling that skeletal muscle to contract until you reach tetanus. Cardiac muscle cells, these contractile cells, do not have that capability. Those are the theoretical maximum contraction rate based on this 200 milliseconds between the stimulus and the arrival of the relative refractory, or the end of the absolute refractory. So if we take a look and compare that from cardiac muscle to skeletal muscle, we can see why tetanus cannot occur in cardiac muscle, but it can occur in skeletal muscle. So if we take a look at this part right here, this is key, because take a look. Right at that barrier between the absolute refractory period and the relative refractory period, the cardiac muscle cell has already started relaxing when it's in the absolute refractory period. When we take a look at our skeletal muscle, it's still in the contraction phase when we are through both refractory periods. So we can keep firing, keep firing, keep firing, and the muscle, we can do contraction and tetanus. We can maintain tetanus if the neurons are firing fast enough. The frequency of stimulation is fast enough, we can reach tetanus. That is impossible with cardiac muscle cells, okay? Which is pretty cool. Okay, so now that we understand kind of a slight difference between neuron action potentials and the new knowledge of our cardiac contracting cell action potentials, with these muscle cells actually contracting in response to the electrical impulses that are coming through, we have to take a look at the sequence. What sequence of events happen when these muscles are being triggered to be contracting? So we're going to start here at time 0. It's a cycle, but we got to start somewhere, right? So we're going to start at the beginning of atrial systole. So this is, systole means contraction. And diastole equals relaxation, right? So those are just general terms. So systole is contract, diastole is relaxation, and we can apply it to all of the chambers. So we're going to start with atrial systole first. So atrial systole is the contraction of the atria. So we can see the little green arrows are the pushing down. So that's the SA node fires, intranodal pathway. This is kind of like right after the P wave in the ECG, and the atria are contracting. Blood is being pushed from the atria into the ventricles, okay? And that lasts about 100 milliseconds. Okie dokie. So then our atria are filled, okay? And then the whole rest of the time, not our atria, sorry, our ventricles are filled because the atria are squished. They're pumping the blood from the top, that last little squirt of blood, and our ventricles are all filled up. Now, if we're going to just keep looking at the atria, this whole rest of the time, the atria are not contracting. They're in atrial diastole. So in this whole 800 milliseconds of a typical cardiac cycle, 100 of it is spent atrial contraction, 700 milliseconds are spent relaxing. Now, they're not totally relaxing, they're being filled with blood. Blood is continuing to come in. So we'll have oxygenated blood coming in from the lungs, deoxygenated blood coming in from the vena cava, and they're just going to keep filling even though the ventricles are off doing their own thing. They're just not actively contracting. Okay, so atrial systole, squish, ventricular, they were all in ventricular diastole, right? So here's ventricular diastole is this tan line. So while the atria, when we started here, when the atria were contracting, the ventricles were still relaxing. We can kind of see that because it's all kind of puffed out like that. So those are our relaxed ventricles. Now, atria is done contracting, the signal goes down, AV node, bundle branches, Purkinje fibers, the signal has been sent to our ventricles. The ventricles are going to start to contract. They contract from the bottom up because remember that twisted muscle pattern, kind of that like you're wringing a towel. So it's twisting and the pressure is coming in from the ventricular walls. Okay. Now pressure is building because the walls are pushing on the blood. It's a blood-filled chamber. When that pressure builds up enough, we can see that those AV valves are going to close, right? They were open when the ventricles are filling, but when the contraction happens, they're going to close. So now our AV valves are closed, but also take a look at our semilunar valves, these guys right here. So semilunar valves are also closed. So there's a very specific point in time where as the ventricles are relaxed, your semilunar valves are closed, but the AV valves are open. When the ventricles start to contract, they produce enough pressure to close the AV valves, but not enough pressure to open the semilunar valves. So this is a period that's called, oh, it's not listed on here. I'm going to erase that real quick so I have some room to write. So this is a period in time that's called isovolumetric contraction. Iso meaning the same, volumetric meaning volume. So it's the same volume. So we're not gaining any blood into the chamber. We're not pumping any blood out of the chamber. And it's during the very early stages of this ventricular contraction. So isovolumetric contraction happens while the AV valves close and the semilunar valves haven't opened yet. Okay, but then the pressure keeps coming. So the pressure keeps coming. It's pushing up against the chambers. Now the chambers are generating enough pressure to actually open our semilunar valves, and this is usually called ejection, the late stage ventricular diastole. The blood is moving out of the ventricles. Okay, so that lasts about 270 milliseconds. So this is our ventricular systole starting at time 100 milliseconds, ends about 370 milliseconds. Okay, this whole time atria has been relaxed, but they're passively filling. Okay. All right. So now, after the ventricles are done contracting, they're going to start relaxing. Atria has been relaxed this whole time. Ventricles are going to start relaxing. So right as the ventricles start to relax, those semilunar valves close, right? Aortic valve and pulmonary valve close because as the ventricles are relaxing, blood wants to pour back down from those tight blood vessels to go into the more loose ventricles. So that catches those cups, closes the semilunar valves, prevents blood from going back into the ventricle. However, the ventricle isn't relaxed enough to open the AV valves. So here's another point in time in this cardiac cycle where all four valves are closed. And this is called isovolumetric relaxation. So again, it's another point in time where blood is neither entering or leaving the ventricles, but the ventricles are in the process of relaxing. It's relaxed enough to close the semilunar valves, but it hasn't relaxed enough to open the AV valves. But it'll continue to relax, and eventually your AV valves will open. All the blood that had been passively coming into the atria can now pour down into the ventricles, and blood continues, and so this is your passive filling. So all the way from here up to here is passive filling from 370 to, a little bit over half. So 430 milliseconds, you have blood just pouring through the relaxed atria down into the relaxed ventricles. So it's kind of interesting. And in this average cardiac cycle of about 800 milliseconds, half of that time, neither chamber is contracting. It's just a passive filling. So you have about 300 milliseconds of time to do all of your contractions, and everything else is just passive filling. Okay, so those terms are really important. The atrial and ventricle systole and diastole, isovolumetric contraction, isovolumetric relaxation. Also that knowledge about when the valves are open and closed, that's going to come back. So, you know, having that foundation when we went over the valve structure is important for this cardiac cycle understanding. So this next slide is one of taking that cardiac cycle, and instead of looking at it in a circular chart, we're going to look at it in more of a linear chart and kind of map everything out. But this picture from your textbook has a whole bunch of stuff on it. So to avoid overwhelmingness, because I know all this stuff is quite overwhelming, I'm just going to take one piece at a time, and hopefully that will clarify and answer questions as we work through this. So the first comparison is they are comparing that cardiac cycle. So again, this is the cardiac cycle, those terms. Systole and diastole. Atrial diastole, remember, was about 100 milliseconds. Oh, sorry, about 100 milliseconds for systole, about 700 milliseconds for diastole. And then it follows again, systole for 100 milliseconds, and then diastole for 700 milliseconds. And then we have the ventricular systole for, was it about 200 and something milliseconds, and then 300 and something milliseconds, or 400 for diastole. So that's your cardiac cycle, these terms right here. Okay. How does the EKG line up with those stages? Well, P wave, remember, P wave was the depolarization of the atria. And right, if we're to draw that line, if I were to draw it better straight up and down, the contraction, which is a systole, starts about halfway, right at the peak of that P wave. Just like ventricular contraction starts at the peak of the QRS complex, kind of at the R, okay? And then our relaxation, ventricular diastole, starts after the heart, the muscles have been repolarized. So that's at the end of the T wave. So lining up your P, QRS, and T to the contraction shows you that the contractions start a little bit after the signal has been sent, the P and the QRS, and the relaxation starts at the end of repolarization. Okay. All right. This next part, this is the one that's a little bit more tricky. So we're going to take one at a time, each one of these lines at a time, and see if we can understand and walk our way through why these graphs are the way they are. So I'm going to take a look at, I'll use purple because that's not any colors on here. The left atrium line first, that's the blue one. Now this is only showing the left side of the heart, but realize this is going to be mirrored on the right side of the heart. The pressures just may not be as great. They would have a different scale on the y-axis, but the pattern would be the same. So left atrium, right? So we'll start here. This is systole, atrial systole, and then the rest of the cycle is diastole. So this is pressure. Millimeters per mercury is the unit of pressure. So between steps kind of 1, 2, and 3, 1 is atrial contraction begins. And take a look at the pressure. It's going up a little bit, right? There's a little bump. Makes sense. If your blood inside the atria and the atrial walls are starting to contract around you, pressure is going to increase. And so we can see that in that little blurb. Okay. And then we have atrial diastole the rest of the time. So this is all relaxation until about right here. And what's happening to the pressure? Well, it's kind of squiggling around a little bit. It goes up just a tad right when the valve closes, which kind of makes sense because the AV valves are open and then the ventricle starts to contract and the AV valve flaps up. That might, you know, produce a little bit of a pressure wave. And then it kind of goes back low, but what's happening? This is all passive filling, passive filling, passive filling. So we're getting up because the blood volume is filling up the atria, so the pressure is going up. That kind of makes sense. And then we have the AV valve open. So we were in this chamber during atrial diastole relaxation, and then all of a sudden the ventricles relax so much that we can open our AV valves. So the pressure is going to go down. It's going to match the ventricle because it's basically one open chamber at this point. AV valve is open, atria and ventricle are kind of open to each other, and then we are going to build our pressure because more blood's coming in. So this is our passive filling to right here, and that's the end of atrial diastole. So we get a little blurb of pressure here at kind of step 2 when atrial contraction is happening. We get a little blurb of pressure here when the AV valve is opening, and we get a gradual pressure increase as blood is filling into these chambers. Okay, so that's the atrium. Okay, kind of makes sense, matches along with the activities of the cardiac cycle. So now we're going to do the red, which is left ventricle. I'll use a green color. So the ventricle is going to follow, that's this red line here. I know I'm using green, but I don't want to put two red lines on there. So at this point, atrial and ventricular are both in diastole. So their pressures are going to be very similar. But take a look right here. AV valve closes. So AV valve was open in kind of steps 1 and 2. Ventricle starts to contract. That's your ventricular systole, right? That's this line right here. Step 3. Okay. Ventricles start to contract, AV valve closes, okay, and pressure is building in the ventricle. So it makes sense, excuse me, it makes sense that the pressure in the left ventricle is going to build rapidly, and it's building rapidly. It's going straight up on this chart. But take a look, AV valve closes here, aortic valve opens here. So it is this period of time that is your isovolumetric contraction that we saw. And it's not a very long period of time. The time scale is all the way down at the bottom, but if you're to track that little tiny sliver, it's a fraction of a millisecond of time between the AV valve closing and the aortic valve opening. Okay, so ventricle pressure is building, it's building, it's building. It's building so much that at this point here, that's when the aortic valve opens. So now the left ventricle and the aorta are basically an open chamber to each other. So that's why we see their pressures are about the same. Okay, and then at this point here, step 6, this is when ventricular diastole happens. So now the ventricles are starting to relax. They relax enough to close the aortic valve. So that's the little cups catching the blood, closing. And then right here, left valve, AV valve opens. So from this point down to this point, this is our isovolumetric relaxation. It's a little bit longer than the contraction time because it takes a little bit slower for the relaxation enough. So aortic valve closes as the ventricle is relaxing, so no more blood can come back in from the aorta or the pulmonary trunk, but we haven't relaxed the ventricles enough to open it for the blood to come in from the atria. And that's what happens here at step 8. So now all the blood that was filling into the atria, AV valves are open, we can now allow the blood to flow in, and we are matching that atrial pressure, just like we saw before. Okay, so that is the ventricle pressure curve. Now, if we take a look at the last line, which is a black one, this tracks the aorta. Now, the pressure in the aorta starts high, right? I just crossed it out. It was on 90 millimeters of mercury. That is your blood pressure, right? So blood pressure is typically 120 over 80-ish. So this is kind of what we're seeing. Where did those numbers come from? The systole contraction pressure over diastole, contraction over relaxation. So right now, the ventricle is in relaxation at this beginning phase, right? So the aortic pressure is relatively low. Ventricle starts to contract. Push, 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 pressure, 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 pressure. AV valve opens. Now we have high pressure in our aorta because the blood is just shoving from this large ventricle space into this smaller tube. And so this is our peak pressure. That would be like our 120 reading, like 120 over 80. That's our diastole or systole is the 120. When the ventricle starts to relax, right here, the aorta pressure goes down because it's matching the same pressure in the ventricle. And then the pressure in the ventricle goes down so much that blood in the atria would rather go back down into that low pressure ventricle, but it catches those AV valve or the semilunar valves, preventing the blood from going back into the ventricle, which is a good thing because we want this one-way flow. So then this blood pressure kind of goes down because it's during that interval of both ventricular and atrial diastole. And then you're going to just hit the next heartbeat and the pressure in the aorta will go up again. Okay, so hopefully that's sitting a little bit better for you, walking through those each line by line, comparing it to the systole and diastole cycles of the cardiac cycle. The last thing is the volumes, right? So we did ECG comparing to the cardiac cycle. We did the pressures comparing to the cardiac cycle. And now we're going to do the volumes, which will lead us into our cardiodynamics, which is the last video of this chapter. All right, so when we talk about volume, we're primarily focused on ventricular volume. We don't really care so much about atrial volume. So this is just left ventricular volume, which would be the same for the right. They have the same volumes that they deal with. So here we're going to track kind of this first step. This is atrial diastole. So this is passive filling, right, from the previous cycle. From 2 to 3, this is our atrial systole. So the volume in the ventricle, so remember, this is ventricular volume. So the volume in the ventricle is going to get that last squish of blood from the atrial contraction at the beginning of the heart cycle. And so now we're at our peak ventricular volume. We call this end diastolic volume, or EDV, because again, it's ventricular volume. End diastole, it's the end of ventricular diastole volume. In this case, we're about 130 mils. Okay. All right, now we do the contraction, right? So that's all timed, kind of steps 3 through 6-ish. So now we're pumping all that blood out. So you can see this rapid decline in blood volume of the ventricle because we're just ejecting it out into the aorta. So what's left is called end systolic volume, ESV. And in this case, it's about 50 mils. So you start off with, when your ventricular is as relaxed and as full as it can be, it has 130 mils. Then you contract, squish, squish, squish as much as you can during that ventricular systole, and you are left over with about 50 mils. Okay, so you don't pump it all out. It's not 100% pump. So then we have what's called stroke volume. Stroke volume is the amount of blood that actually gets pumped out in each contraction. So here's our stroke volume. So stroke volume is EDV minus ESV. It's what you start with, subtract by what you end with. So this would be 130 minus 50. So stroke volume in this example is 80 mils. So I did my math right? Yeah. So every heartbeat at this particular contraction, force of contraction, heart rate, all that kind of ventricular length of time for ventricular systole, every time this particular heart beats, it's going to send out 80 milliliters of blood per pump towards your systemic circuit and towards your pulmonary circuit. All right, so those are cardiac cycle, pressures, and volumes all kind of lined up. The last slide in here is going to be talking about heart sounds, but before we move on, this understanding of EDV, ESV, and stroke volume is the foundation of what we're going to start with on the last video dealing with cardiodynamics. All right, so on this slide, it's showing us kind of the same, the pressure changes piece from the last graph, lined up with our heart sounds. Now there's 4 heart sounds listed here, but there's really the 2 major ones that we are familiar with, the lub dub, the boom boom, boom boom, boom boom. Those are S1 and S2. They're associated with, first, the lub is the AV valves closing. Let me double check to make sure. Yes, it's the loudest sound. It's the lub. Dun, 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 dun. It's the first one. So that's the AV valves closing right here. This sound, lub, lub, lub. Both of them together. So it's not one or the other. It's both left and right. AV valves are closing at the same time. Lub. And then the dub sound is the closing of the semilunar valves. Okay. So it's lub, dub, lub, dub, lub, dub. So the heart sounds are the closing of the valves. AV valves first, when the ventricle is contracting, preventing blood from going back up into the atria. The dub is the semilunar valves closing, preventing the newly ejected blood from going back into the ventricles. That's your lub dub. The other sounds, the S3 and S4, are caused by blood flowing into the ventricles. So S3 is blood going into the ventricles, and S4 is atrial contraction, kind of right here. both quieter. You'd probably have to have a little bit more practice and experience trying to identify those sounds, or if something might be going wrong with those blood flowing or contracting of the atria. We will have an opportunity to have some stethoscopes, and you can listen to your own heart sounds if you haven't done so yet. We'll have that opportunity, and just think about what's happening. Closing of the AV valves, lub. Closing of the semilunar valves, dub. And I'll have posted some videos and animations of this process as well. All right, so that wraps up our cardiac cycle physiology lecture. I will see you next time to wrap up with cardiodynamics. Bye.